Welcome to Click for IB Biology. This playlist is relating to neural signaling and the nature of nerve impulses. In this particular video, we're going to be looking at nerve structure and the development of resting potentials. There's a PDF of my presentation that's down in the description below. There you will also find 10 questions with answers, and you can use those in order to check your understanding. So let's look at the structure of nerve cells and their locations. So the human nervous system is divisible into two major components. There's the central nervous system and the peripheral nervous system. The central nervous system is composed of the brain and the spinal cord shown here. So that's part of the central nervous system. Emerging from and connected to the brain and the spinal cord are sensory and motor neurons of the peripheral nervous system. Now, these are actually divisible themselves into two particular types, known as the somatic and the autonomic. So autonomic is self-explanatory in a sense that it's the automatic features which we are not consciously deciding, such as the contraction of smooth muscles in the gut or in our blood vessels. But over here we've got the motor nerves and the sensory neurons of the somatic system, sensory neurons delivering information coming into our central nervous system, and motor neurons delivering instructions to muscles for our response. So as we go forward, we'll take a quick look at cells of the central nervous system, motor neurons in this video, and later in another video we'll take a look at sensory neurons. Moving on to the structure of nerve cells within the central nervous system, the neuron structure and their development. So these cells are relatively small, approximately 5 micrometers in diameter. And as you can see from the image, they have this star-like shape where these extensions of the cell membrane with cytoplasm inside are called dendrons. And these indeed themselves end up branching into finer structures called dendrites. And it's actually in the dendrite which makes the synapse connections to another nerve cell, or indeed to a muscle. But in this case, in the central nervous system, it's largely neuron 1 connecting to neuron 2 at synaptic connections which occur between the dendrites. And so on one side of the dendrite, if we explore this at a greater resolution, at one end of the dendrite you would have the synapse structure. As the impulse comes from the cell body, travels through the dendron into the dendrite and results in a chemical transmission across from the synaptic junction here, across the synaptic cleft. That's the space, which itself is between 20 to 40 micrometers, very, very small diffusion distance, and it makes connection with the membrane of the second neuron. Now, we describe these as presynaptic membranes and postsynaptic membranes. And the postsynaptic membrane differentiates and introduces into its membrane structure receptors, and those will be explored in topics such as cell signaling, and indeed in this particular topic in future videos we're going to be looking at. Now, when the cells of the nervous system communicate with each other, they set up networks. Perhaps cell 1 is connected to 2, is connected to 3, and we have synaptic transmission between 1, 2, and 3. And so our network is 1, 2, 3, with some kind of initial sensory input here, and perhaps some kind of final motor output here. So we have input and output in this form. As they begin to signal with each other, the connections that they form between the nerve cell become strengthened with the addition of receptors, but also because of a process called neural pruning, in which other connections are broken down, including whole nerve cells are eliminated from this network so that the network becomes more refined. And although this occurs as we go through the process of learning, the interesting thing is that the capacity to form these networks is retained throughout our lives and is an example of neuroplasticity, where we can learn things later in life, but we can also at any stage in our life learn new things. So these are typical cells of the central nervous system. But if we move to the motor neurons of the peripheral system, so this is an example of a motor nerve. You can use the word nerve or neuron, and neuron can also be spelled in some books with an E on the end, which is the British spelling. And so here you can see the similar structure with the dendrons, the dendrites, the soma, which is the alternative name for the cell body. But here, one of the dendrons has become elongated into what we call an axon. And in a motor nerve, this can be as long as 1 meter, such as the nerve emerging from the base of your spine in the central nervous system and traveling all the way down to muscles in the foot region. And they can be, the axon therefore is about 1 meter long. So this is a long extension of cytoplasm with membrane around it, ending in the axon terminal and synaptic neuromuscular junction, which would occur down here. Now, the additional feature in vertebrates, and indeed ourselves, is that this axon is wrapped with what are called Schwann cells. Now, the Schwann cell, as shown here in this micrograph, there's the axon plasm with the axon membrane like so, and this Schwann cell has developed over time, and during our embryonic development and our fetal development, the Schwann cells grow around in a series of growing round and around and around like this, leaving this image here like so. And what this does is insulates the axon from the extracellular fluid in these regions here. So it's insulated from the extracellular fluid by the presence of fat stored within the Schwann cells wrapped around the axon. However, the insulation is not present where the Schwann cells meet, and this creates a structure called the node of Ranvier. Later we will discuss the significance of these nodes in saltatory discussion. Now, researchers have placed electrodes into the axon, and also with reference to the external tissue fluid, and when connected to an oscilloscope, we're able to record that the charge inside the axon is recorded at minus 70 millivolts, and we call this the membrane. This is a membrane potential, a charge across a membrane, and it's sitting at minus 70 millivolts, and unless a nerve impulse arrives, it will remain so. So we call that the resting membrane potential, usually shortened to resting potential. Now, the nature of a nerve impulse is actually a flow of positive ions, and in particular, the flow of sodium ions, which flow as a wave along the inside of the axon from the cell body all the way down to the neuromuscular junction, where it will trigger a muscle contraction. And so our study now will return to the beginning of this, and how does this sodium wave begin the initiation of the nerve impulse, and then later we'll look at the synaptic transmission to a neuromuscular junction in a different topic of muscle contraction and cell signaling. The next video looks at how the resting potential has been established. There's a link to the channel and the playlist, of course, for your convenience.